Evening, ladies and gentlemen. Thank you very much for giving me voice. Today, I'll be talking about comparative study published in recent number of environmental science and pollution research under leadership of Politecnico Torino Professor Deborah Fino. The work is titled Comparative Study of Polyphenol Extraction Using Physical Techniques and Water as Solvent, a Sustainable Approach for Valorization of Apple Pommes. Apples, among most cultivated fruits, and 65% production is transformed into apple juice, generating an amount of waste named apple pommes, which includes seeds, skin, and other components. Disposal of these products is waste, and pommes is a region of polyphenols that can be used, utilized, and active as cosmetic, nutraceutical, or pharmaceutical products. Water is used as extraction solvent in thermal steel extraction, ultrasound-assisted extraction, and microwave-assisted extraction. These are some industries which are suitable for that. These apples are one of the most consumed fruit around the world. In Europe, the main producers apples are Poland, Italy, and France. Approximately 12 million tons is yearly produced in Europe. Approximately 71% are consumed in the fresh ones, while 20% is going to yield value-added products. 65% of apples exported into of this 20% is transformed into juice concentrate, with the remaining portion being utilized in the production of various items. Package of really safe apple juice, apple cider, vinegar, vermouth, apple tripuris, jams, dried apple. Next, the processing methods mentioned above result in the production of waste, specifically apple pommes, which includes seeds, skins, and other components. This waste typically accounts for 20 to 30% of the initial apple weight, a proportion that varies depending on the apple variety and the processing technology employed. Discarding these byproducts directly in the environment can have several dangerous impacts, as the production of greenhouse gases and contamination of underground water table. They can represent a source of secondary pollution, such as emitting a foul smell due to microbial attack, and also acting as human disease vectors. The effective management of apple pommes requires strategic approaches to minimize environmental impact. This government for biopharmaceutical processes. The apple pommes contains large fat numbers of compounds that, such as polyphenols, dietary fibers, and fatty acids and minerals, can be used as active ingredients in cosmetic and nutraceutical. Ultraviolet radiation induces skin damage and photoaging and flavor. Liquid, and solid liquid extraction techniques are commonly employed techniques use friendliness, solvents, alcohols, methanol, ethanol, acetone, diethyl ethyl. The recent studies recommended the use of innovative technologies in increase of use of bioactive compounds and to improve the efficiency of the production extraction. Ultrasound-assisted extraction and microwave-assisted extraction. Microwaves interact with the polar molecules of the solvent, generating a rapid and uniform heating of the materials, while ultrasound, a phenomenon of cavitation enables faster transfer of molecules from the matrix. In the literature, there are no studies comparing these two techniques, extraction and pomace. This present study aims to describe the compare of these two physical methods to identify the pomace extraction of polyphenols. From apple pomace, toxicity, reliability, safety, and ease handling. The polyphenol extraction methods were investigated to consider both the efficiency of the process. Environmental analysis was performed using life cycle assessment. Apple pomace samples were kindly supplied by Agriculture Corporation of Bibiana, and red delicious crimson, Granny Smith, and Gawel varieties were most. Fresh apple pomace was later obtained by byproducts from side bioproducts production. The operation was vortex using relatively low 50 degrees drying temperature to preserve bioactive compounds. Gallic acid and sulfur were standards acquired for sigma-aldry. Methyl phosphate, chochaltum, reagent sodium carbon, and sodium made for milk. Water extraction was distilled water obtained from the laboratory distillation apparatus. The approximate composition of pomace was determined using pass of an air oven to 103 degrees. For ash determination, 0.5 grams of dry samples were heated in muffin furnace for 6 hours and the residual were weighed. Lipids were determined using artificial, where 5 grams samples are treated with petroleum ether in the substrate apparatus protein contained. Once determined uses the Dumas method, where CMS analyzer was used to determine the nitrogen. Water extraction was an optimization strategy for this study. A total of 27 screening experiments were conducted for each extraction method. Solid to liquid ratio, temperature, of power were the most significant extraction parameters. Nevertheless, to gain a more comprehensive understanding of the mechanism and additional series, this set of experiments be referred to as the thermal stirred extraction. These stirring conditions, as well as the type of solvent, were also kept constant throughout the experiments. The solid-liquid ratio was varied between 110, 120, 130 with every experiment. For the extras was added to appropriate volume in beakers based on the solid to liquid ratio of experiment. Magnetic steel bar was inserted to ensure solution homogeneity during the extraction done by stirring the system. The experimental setup included an hot plate stirring with control. The diluted sample were placed inside an ultrasonic bath positioned above the plate. The connected to a probe 30 mm was immersed 2 cm deep into the beaker component. The microwave assisted extraction was carried out in microwave digestion. Waters were holding consistently high pressure reaction vessels, showing high sample throughout and high. Was filtered using folded qualitative paper. Filter Waitman crate, particle mesh vacuum tubes. The total polyphenol contents, the apple pomace samples, was determined. Standard procedure outline was within the sodium carbonate was added. After incubation at ambient temperature in the dark for 1 hour, the absorbance at 1765 with UV spectrophometer was measured. The results was as milligram equivalent. Cynetics was conducted to study the extraction. Kinetics analyzed time-dependent evolution polyphenol concentration extracted from apple pomace. The study kinetic was process of kinetics proportional to the time and great constants. This life assessment was using life cycle assessment to norm of 2006. We investigated type of ISO 13044 and extraction scope the identification of lower impact The approach was grade-to-grade in a processment adopted to determine the product. What extraction performance was fast. This was three-step: collection and transport, refrigerating, calculated the effect. Then was water extraction, transported output, transported water, energy for gas reaction, output were polyphenol solution, wastewater. While the advanced assisted extraction, apple refrigerated stores. This was screening technology was ratio. Polyphenols increase the disruption of the cellular structure of the matrix, increasing the permeability of the cell mix. These screening were identical and third solution. Extraction time was evaluated for miles according to the results obtained from screening. This extraction were life cycle assessment depicts that all extraction techniques have similar trends in both climate change category and energy demand. This microbial extraction operation was higher in components water compared to water-based extraction. Increased pomace to water ratio salted the biggest reactor working volume, the more wastewater extraction, based on Sprucebark and acetanol. This most confusing assumption plays critical role. The renewable and fossil were water-based were mostly, where unfortunately not renewables was 0.22. So this water is 90% was still in fossil, so not very friendly. In this study, ultrasound assisted were screened, and the best stage achieved for the apples was ultrasound assisted was 1 to 2 for 67 10 minutes, while the best were 300 volts, but less energy and less efficiency of 80 to 70%, 80 to ultrafiltration. So that's all what I wanted to say for today. Thank you very much for giving me voice.